So here we're talking about the animal cell. First, we start off with the nucleus, which makes RNA. The chromatin actually makes chromosomes, and then we have the nucleus, which is called the control center of the cell. It contains DNA where transcription occurs. Next is ribosomes, which are used for protein synthesis. The lysosomes are used for the breakdown of waste from the cells. The rough endoplasmic reticulum assembles proteins, and it's called rough due to the ribosomes embedded in it. The gluteus apparatus packages proteins in vesicles for transportation. Microfilaments, intermediate filaments, and microtubules all make up the cytoskeleton. Smooth endoplasmic reticulum synthesize lipids. Mitochondria produce energy from cellular respiration and is also the location of an electron transport chain. Vacuoles are just space to hold material. Cytoplasma is the plasma that's inside the cell. The plasma membrane holds everything inside, like the cytoplasma. Perosomes hold reducing enzymes. Centrosomes hold spindle fibers for cell division. Secretory vesicles release material through the plasma membrane. Let's talk about the nucleus. The nuclear envelope is very important, and it allows what goes in and outside, and the nuclear limitides are fiber proteins that hold the structure. The nucleus, as we stated earlier, makes RNA, and ribosomes are made here. Spliceosomes are made from sRNA and proteins to splice the introns in the mRNA. This is a process of RNA processing. The more active the chromatin is, the easier it is to get genes to express them. Euchromatin are more active and heterochromatin are less active. Nuclear pores are for transportation of molecules through the nuclear envelope, like RNA or ribosomal proteins. They're very important to help harmful material. The next part is the mitochondria. The outer membrane is made out of phospholipids and contains pores to let materials in and out. It's permeable for some nutrients and ions. The inner membrane is folded to increase surface area and helps with ATP production and is only permeable to oxygen. Inner membrane space is used for the electron transport chain and is the same composition as the cytoplasm. The matrix is a mixture of the proteins and enzymes to help with the synthesis of ATP. The cristae is a folded in the intermembrane. The ribosomes are used to translate mitochondria DNA and proteins. It has its own ribosomes and it only works for the mitochondria. The granules create a place between inner and outer for enzymes to work. ATP synthase particles is what is used to make ATP. In a mitochondria DNA, it has its own DNA and replicates itself. The mitochondria and chloroplast are both related to the bacteria. Evidence: they have their own DNA, they self-replicate, and they produce their own proteins and enzymes.
Plant cells are eukaryotic cells that differ from the cells of other eukaryotic organisms in several ways. One distinctive feature of a plant cell is its chloroplast. The chloroplast is the organelle of photosynthesis and captures light energy from the sun and uses it with water and carbon dioxide to make food for the plant. The plant cell is structured so that the chloroplast is enclosed by an inner and an outer phospholipid membrane. Between these two layers is the intermembrane space. The fluid within the chloroplast is called the stroma, and the stroma contains one or more molecules of small circular DNA and also has ribosomes. Within the stroma, there are stacks of thylakoids, which are suborganelles that are the site of photosynthesis. Photosynthesis takes place on the thylakoid membrane, and within that membrane is the complex of proteins and light-absorbing pigments such as chlorophyll and carotenoids. These two specific pigments allow the capture of light energy from many different wavelengths because both capture different wavelengths of light, and the chlorophyll is what gives the green color. The next feature of a plant cell is the peroxisome. The peroxisome is a membrane-bound packet of oxidative enzymes which break down the hydrogen peroxide into water and oxygen and also convert fatty acids to sugar. The peroxisome also assists chloroplasts in photorespiration. The mitochondria is formed by rod-shaped organelles, and it is considered the power generator of the cell, converting glucose into ATP through the process of oxidative phosphorylation. The mitochondria is a double membrane-bound organelle and has an outer and inner membrane made up of phospholipids and proteins. The mitochondria is also involved in signaling, cellular differentiation, and control of the cell cycle and growth. The cell wall is the rigid layer found outside the cell membrane, which surrounds the cell and contains cellulose, protein, and other polysaccharides. The cell wall provides structural support and protection for the cell, and the pores in the cell wall allow water and nutrients to move in and out of the cell. The cell wall prevents the plant cell from bursting when water enters the cell. The nucleus is enclosed in a double membrane and communicates with the surrounding cytosol through many nuclear pores. Within each nucleus is nuclear chromatin containing the organism's genome. The nucleus stores cells' DNA along with coordinating metabolism, growth, protein synthesis, and reproduction, also known as cell division. The nucleus is about 10% of a cell's volume and a highly specialized organelle which serves as the information and administrative center of the cell. The nucleolus produces ribosomes which move out of the nucleus and take positions on the rough endoplasmic reticulum where they are needed for protein synthesis. The nucleolus is composed of protein, DNA, and RNA, and functions as a long ribosomal RNA molecule, which is transcribed from DNA at the nucleus, being processed into 3 mature RNAs packed together with certain types of proteins to create a small and large ribosomal subunit. Once these small and large ribosomal subunits are created, they are transported out of the nucleolus for the cytoplasm to use in functions such as protein synthesis or translation. Ribosomes are packets of RNA and protein that play a crucial role in the formation of plant cells. They are the site of protein synthesis, and they are comprised of both large and small subunits. Ribosomes are produced and assembled in the nucleolus and the subunits that are also formed there are only separated when the production of protein synthesis is not being carried out. The units of these ribosomes will leave the nucleus through nuclear pores and unite once again in the cytoplasm for protein synthesis. The nuclear envelope is a double-layered membrane that separates contents of the nucleus from the cellular cytoplasm. The nuclear envelope is covered with holes called nuclear pores that allow specific types and sizes of molecules to pass back and forth between the nucleus and cytoplasm. The nuclear envelope is attached to a network of tubules where protein synthesis occurs, which is the endoplasmic reticulum. The cytoplasm provides mechanical support to internal structures and is known as the medium for suspension for internal organelles of the cell. The cytoplasm is the site of many biochemical reactions such as glycolysis and protein synthesis. The cytoplasm is also the place where all cells expand and the growth of the cell happens. It aids in the movement of different cellular elements, and the enzymes here metabolize the macromolecules into small parts so that it can be easily available for other cellular organelles like the mitochondria. The cytoplasm works as a means of transport for genetic material and transports the products of cellular respiration as well. The cytoplasm acts as a buffer and protects the genetic material of the cell and cellular organelles from damage caused due to movement and collision with other cells. The plasmodesmata are small channels that directly connect the cytoplasm of neighboring plant cells to each other and establish living bridges between the cells. They penetrate both the primary and secondary cell walls to allow certain molecules to pass directly from one cell to another, which is very important for cell communication. The plasmodesmata is lined with the plasma membrane, so all connected cells are united through one continuous cell membrane. It's widely thought that the plasmodesmata, by constricting and dilating openings at the ends of this, plant cells are able to regulate the passage of small molecules such as sugars, salts, and amino acids. The rough endoplasmic reticulum appears pebbled due to the presence of numerous ribosomes on its surface. The proteins synthesized on these ribosomes collect in the endoplasmic reticulum for transport throughout the cell. The smooth endoplasmic reticulum appears smooth and helps function the plant cell through lipid and steroid hormone synthesis. The Golgi apparatus is surrounded by several small membrane-bound vesicles and composed of numerous layers of fluid-filled membrane sacs. These sacs are stacked near the outer layer edges of the endoplasmic reticulum, which is near the nucleus. The Golgi apparatus functions to process, modify, and sort newly produced proteins that will have arrived from the endoplasmic reticulum. By modifying these proteins, an example would be adding and deleting certain molecules of sugar to change branch sugar structures found on the newly formed proteins. The plasma membrane is the structure of the plant cell that forms a semi-permeable barrier between the interior of the cell and the external environment. The plasma membrane functions in transport of molecules into and out of the cell and contains proteins and other molecules essential for receiving signals from the environment and from plant hormones that direct plant growth and division. The plasma membrane is provided its structure through lipid molecules, and the most abundant of these lipid molecules are phospholipids. The smallest membranous vesicles are small membrane-bound sacs, and they function in cellular storage and transport. Its membrane is capable of fusing with plasma membrane and membranes of endoplasmic reticulum and Golgi apparatus. These vesicles store and transport substances from one cell to another or from one part of a cell to another part of a cell. When these vesicles fuse with the plasma membrane and membranes of the endoplasmic reticulum and Golgi apparatus, they do this so they can release contents outside of their boundaries of the cell. The leucoplasts do not contain any pigments and are located in the roots and non-photosynthetic tissues of plants. Leucoplasts may become specialized for bulk storage of starch, lipid, or protein, but in many cells, they do not have major storage function at all, and instead they make molecules such as fatty acids and many amino acids. The filamentous cytoskeleton is made up of filamentous protein and provides mechanical support to the cell. The cytoskeleton is composed of microtubules, actin filaments, and intermediate filaments, which each help give shape and organize the cell parts and provide basis for movement and cell division. Bacteria cells are prokaryotic cells lacking membrane-bound organelles and well-defined nuclei. One of the most distinct features of a bacteria cell is its cell wall. A bacteria cell wall is defined as a peptidoglycan cell wall composed of disaccharides and amino acids, giving bacteria structural support. A peptidoglycan cell is made from polysaccharide chains cross-linked by unusual peptides containing D amino acids. A cell wall in a bacteria cell is different because it can split into two different cell walls, one known as the gram-positive cell wall and one known as the gram-negative cell wall. These are differentiated by reaction of cells to the gram stain, and each have their own features. So for the gram-positive cell wall, it's a thick cell wall composed of peptidoglycan, lipids, and teichoic acid, while the gram-negative cell wall is a thin cell wall composed of peptidoglycan and an outer membrane formed by lipids, proteins, and lipopolysaccharides. In both of these cell walls, peptidoglycan is apparent, and that is what is responsible for the strength of the cell wall. Most have the gram-negative cell wall, and that is the thin cell wall. The next feature of a bacteria cell is the capsule, which is a discrete layer of polysaccharides deposited outside of the cell wall. The type of capsule that we find in bacteria is the glycocalyx, which is a thin layer of tangled polysaccharide fibers and occurs on the surface of cells growing in nature. Capsules rarely contain amino sugars or peptides and have many functions. The two main functions of capsules are to protect bacterial cells from predatory protozoa and to protect cells from the effects of drying. Ribosomes in bacteria cells translate the genetic code from the language of nucleic acid to the language of amino acids. They are also much smaller and have a slightly different composition and molecular structure than they do in other cells. Ribosomes here are never bound to other organelles, but instead are freestanding structures distributed throughout the cytoplasm. The cytoplasm of a bacteria cell functions of cell growth, metabolism, and replication. The cytoplasm here is a gel-like matrix composed of water, enzymes, nutrients, wastes, and gases, and contains cell structures such as ribosomes, DNA, and plasmids. The strand of DNA here is localized by a nucleoid, while everything else in the cytoplasm is spread out. The plasma membrane in a bacteria cell is also known as the bacterial cytoplasmic membrane and is composed of a phospholipid bilayer and has all general functions of a cell membrane. The plasma membrane has a selective permeable barrier that regulates the passage of substances into and out of the cell. The plasma membrane is 40% phospholipid and 60% protein and doesn't allow through large molecules or any charged particles. The DNA, also known as the nucleoid, is not membrane bound and is attached to the cell membrane, which puts it in immediate contact with the cytoplasm. The nucleoid has no uniform shape or size and is composed of multiple compacted copies of DNA in a continuous thread. Most bacteria have a single circular strand of DNA, which is responsible for replication, but some have two or more. Plasmids are also found here, which are smaller circular auxiliary DNA strands. The mesosome is areas in cell membrane that fold inward to maximize the surface area. They play a role in cellular respiration, and they are part of the structure of the plasma membrane, which lines the cell wall. These folds can become distinct vesicles, which can then be used to contain material. The bacterial flagellum is a helical-shaped structure composed of subunits of a protein flagellin, arranged in several intertwined chains. One end of it is free, which is the basal body, and the other end is attached to the cell, which is the hook. The basal body is anchored in the cytoplasmic membrane or the cell wall. The energy for this movement is provided by ATP. The presence of rings surrounded by a pair of proteins, MOT, drive the flagellar motor, causing the rotation of this filament. And another set of proteins, which are the flea proteins, function as the motor switch. This motor switch reverses the rotation of the flagella in response to certain intracellular signals.
The endomembrane system is a system in animal and plant cells that aid in the packaging, modifying, and transporting of proteins and lipids. This is important because this is a system of how proteins are tagged and secreted to go where they are needed in the body or cell. It all starts in the nucleus, where RNA transcribes the DNA. The mRNA is then exited through the pores situated on the nuclear envelope to meet with ribosomes waiting to translate them into proteins. These proteins are then moved through the rough endoplasmic reticulum to be chemically modified, such as adding carbohydrate side chains. The smoothie yard is where these modified proteins are synthesized and detoxified. This is also where calcium ions are stored. Any proteins that leave here are put into vesicles that take them to the Golgi apparatus. The Golgi apparatus is where proteins are tagged by adding phosphate groups and are sorted to leave the cell through exocytosis or to go to another organelle. Vesicles are how the proteins are transported throughout the whole cell. From here, lysosomes are the waste house of the cell. They'll take any proteins that didn't come out right and recycle them. The plasma membrane is the final destination of proteins that are leaving the cell. They will leave through exocytosis from the vacuoles. Here we have the skeleton of the cell, the cytoskeleton. It includes the cell membrane, microtubules, and the filaments. The cell membrane is the outer protective layer that holds everything inside the cell together. The actin filaments are the narrowest of the three. They are a double helix strand of many actin monomers. They're used for movement, like for example in cell division. They are also the paths for transport throughout the cell. Then there's the intermediate filament. These are mainly used for the structure of the cell. Lastly, the microtubules are used for the structure, but are also to be used in the separation of cell and cell division. They are also the paths of transport through the cell if larger chemicals are being